Hello everyone, hope you all are doing well. So today we are going to discuss the next question of our agriculture optional answer writing practice series. And today's question is describe different approaches for improving the characteristics of inbred lines. So inbred lines have been asked previously also, and this question is particularly taken from this year's agriculture optional paper of UPSC Civil Services examination, that is from paper of 2024. So let's see how we can approach the answer. So introduction, the basic point what you should do is to define the inbred lines. So you should also define, and for here in answer writing practice series you should understand what inbred lines are. So inbred lines are homozygous genotypes produced by homozygous genotypes produced by repeated selfing with produced by repeated selfing with selection over several generation repeated selfing with selection over several generations. So what happens for several generations when we keep doing repeated selfing? So obviously when we will do repeated selfing, we will get homozygous. We all know that difference, because in selfing, self pollination, we get homozygous, and in cross pollination we get heterozygous plants. So although you can definition, you can see that we are doing repeated selfing over several generations. So obviously we will get homozygous genotypes, and these homozygous genotypes which we get are only inbred lines. So I hope you understand this, and from here you can also get the characteristics of inbred lines, that these are homozygous. They consist individuals of same genotype. They are maintained by repeated selfing. So these are simple characteristics of inbred which you can simply get from the definition only. So this is what inbred line is. Now let's move to the body of the question, which is the important part, that is what are the approaches for improving the characteristics of inbred line. So we will see that. We will see those approaches. So first of all, we can do cross breeding. Okay, that is hybridization. Cross breeding, or you can say it hybridization. So in cross breeding, what are the things we can do? We can do outcrossing. We can do wide crosses. That is crossing with distant crosses, okay, or distant genetically populations. Okay, distance genetically distant populations, through which what we will get outcrossing and wide crosses, and through which we can introduce some novel traits which are not present in inbreds, such as it could be pest resistance, it could be tolerance to some environmental resistance or anything. So this is what we can do. Secondly, we can do back crossing, the most common method, that is crossing the inbred lines with the parents itself. As we can see here, the name only suggests back crossing, that is we are going back. So crossing the inbred line with parents, so that through which we can get some desirable traits, okay, which were not passed or which will be passed through the parents only. Okay, we can do marker assisted selection. Marker assisted selection. Here we can do genomic selection using some markers. Genomic selection. Okay, here we can also get some beneficial traits. We can do QTL mapping, that is identifying quantitative trait loci. Okay, so what it does, it can also help in development of these inbred lines, and their genetic potential will be increased. So marker assisted selection, we can do genomic selection and QTL mapping. Next, we can do obviously recurrent selection, that is multiple selections, multiple generations of selection. Simply we can say multiple, that is reselection, doing recurrent selection, reselection generation after generation, okay, to get the overall fitness of either it could be yield, disease resistance, or any other thing. Multiple generations of selection. We all know what recurrent selection is, and if you are having problem with the conceptual understanding of what recurrent selection is, you can refer those videos where I have discussed recurrent selection in detail. You will get the detail there. Okay, multiple generations of selection, and I think in static videos I have discussed all these things in detail, so you can refer those only. Or we can make synthetic and composite crosses. Synthetics and composite process. So it involves creating new synthetic varieties by recombining multiple inbred lines. Okay, what we are doing, we will recombine multiple inbred line, and we will develop some new synthetic varieties, and these and from these varieties we will select the superior individuals over several generations. We will breed for several generations, and in that we will develop some, or we will select some superior individuals. So that can be done. We can do induced mutation. Obviously, you can do mutation breeding. That is, through mutation we will try to improve the inbreds. Okay, we all know that here we can use mutagens, and this could be a rapid development method. Polyploidy is another method. In polyploidy we can obviously do chromosome doubling. That is increasing the ploidy level. That is, from diploid to, you will make tetraploid, for example. We will increase ploidy level. For example, there is some inbred is diploid, so to get the desired character we will make it triploid. So this is what we can do. So it will also help us to get better kind of quality. Next, we can do inbreeding depression mitigation. Obviously in inbreds, due to several selections of selfing, there could be problems of inbreeding depression. So what we have to do, we have to mitigate those inbreeding depression mitigation. So it can be done through selective breeding or through hybridization and other things which we discussed earlier only. It can be done through selective breeding or hybridizations, etc. Next, we can do epigenetic modifications. Epigenetic modifications. That is, we can modify the epigenetic factors that influence the gene expression. Okay, so that the gene expression is under our control and we get a better performing inbred line. Okay, for any character, we can do genetic engineering. Simply use of CRISPR Cas9, or we can use gene editing. Simply we can write, we can use gene editing. That is editing of the gene of inbreds for our advantage. So that can be done. We can insert some foreign DNA into inbreds so that we get that quality or that advantage which we want. We can do environmental manipulation. Environmental manipulation. That is, we can improve the growing conditions of inbred. Simply, while growing, if we keep the environmental in the environment, some kind of environmental things in our control, or we can simply improve the growing condition, obviously the quality of the inbred will be better. So that can be done. Next, we can do cryopreservation and germplasm conservation. Preservation and germplasm conservation. Simply what we are doing here, we are preserving genetic diversity, because in inbreds, due to increasing of homozygosity, the diversity is reduced. So what we have to do, preserve the genetic diversity, especially the cases in inbreed where we get some better inbred in some generations. We should try to preserve its diversity so that it can be used later in development of other inbred lines for future breeding opportunities. Simply, so these are the some 10 11 methods through which we can improve the characteristics of inbred line, and I hope you understand all this. And these are simple ones. You have to just remember the some kind of selections and these methods. So that was all. You can simply conclude about writing the importance of inbred in plant breeding methods or any other conclusion which you want. That will be enough. So I hope you understand all the parts of the question. So that is all for today. If you like it, please press the like button. Please do share and please do subscribe. It takes a lot of hard work to make such videos for you on regular basis. And if you like, if you share, if you subscribe, it motivates us to make more such videos for you regularly. So that is all for today. Have a nice day. Thank you.